Well, congratulations, you survived plants, and we're now on to human physiology. And we're going to be on this for a while now. So, this is chapter 35. We're going to start here and keep on going. When we start with the nervous system, we should understand that the nervous system allows you to respond to your environment. It also coordinates your muscle movement and controls many of your bodily functions. And all multicellular animals are going to have some form of a nervous system except for sponges. And we're going to get into this a little bit more now. So when we talk about the nervous system, we need to focus on the types of nerve cells that exist. And when we say nerve cells, we're going to use the term neuron. And we're going to talk about 3 different types of neurons. We're going to talk about sensory neurons. These are ones that can sound like what they do. They sense for your environment. They receive information and then process it and send it to the rest of your body. We're also going to have interneurons. These are the ones that will receive the information from the sensory neuron and then continue to process it and then communicate it where it needs to go next. And finally, we will talk about motor neurons, and these are ones that synapse with your muscles to produce response. So in this course, we really focus on human physiology because we do not have time for anything else. Your book will certainly review other animal systems. I just briefly am going to review the fact that there are different types of nervous systems. So again, sponge is actually very, very simple. They don't really have a nervous system, but they still can respond to chemical and physical cues in their environment. Once you move on to the cnidarians, which is the ones with radial symmetry, they tend to have a diffuse nerve net. And then once we move into bilaterians, this is where we are, they're going to have the more complex nervous systems, and then we're going to see that evolved over periods of time to what we see in modern day humans. And again, early nervous systems, really you just need to respond to whatever is going on in your environment, light, temperature, odor. Even single cellular organisms, frankly, have to respond to light, temperature, and odor. But as you move into being multicellular, then there has to be coordinated movements, also ways to find food, mate, choosing habitats. And again, as we developed more complex body systems and specialized organs, this obviously led to more complex nervous systems. So you can see in this image, we're mostly going to be focusing on bilaterians, and momentarily we're going to shift completely over to humans. But you can see that these organisms here, B through F, these are your bilaterians. And then as in contrast, you can see that your cnidarians, such as the sea anemone, they're going to have a nerve net. But we're going to focus now on the organized nerve system found in bilaterians. You can find that information is transmitted to different regions along what's known as a nerve cord, a bundle of fibers, and all of these animals here shown that are bilaterians have this central nerve cord that runs throughout. It becomes more advanced as you, or more complex, I would say, as you move into upper level or more modern day bilaterians. Again, early bilaterians, which is a more simple system, you have paired ganglia, and this is basically just groups of nerve cells working together. And then later on, you move into organisms that have a brain, and you can see this even in early earthworms all the way through frogs. So this is a more complex centralized collection of neurons able to process information. And then as we get into later organisms in the evolutionary tree, you see that you now are allowing more complex behaviors, and we're going to be talking about memory and learning and even reasoning in some more animals. And we don't get into this in all animals. Again, we're going to focus really on human physiology, but again, you can see how this might have evolved over time, and certainly in other types of courses you can learn more about this. And as we mentioned, as you move into bilaterians, which are all the ones shown here, bilaterians, we talk about having organized movement in one direction rather than sort of grabbing in all directions. Hence, you can divide the body in half only once. There's a head and a tail region. That tended to lend to one organized movement in one direction, which then led to the development of a head, known as the cephalization process. And when that happened, then you start to see the evolution of the brain, and then more specialized sensing organs such as the eyes in one location, going after prey or evading a predator. And you actually see that this evolved many times in different animals. independently, but it did confer an advantage in each situation. Again, you have forward locomotion, ability to be a predator, and also sense your environment and move in a much more coordinated fashion. But as I mentioned, we really don't have time to get into the history of how the nervous system evolves, so we're really going to focus in on the nitty-gritties of human neurology and human function of this nervous system. So when we talk about neurons, we immediately have to talk about how is a neuron comprised, and these are cells that are very different from your average everyday cell. Actually, they can be very long. Some of the longest cells in your body are your neurons, going all the way from your spinal cord down to your toes. And we're going to talk about the basic composition of any neuron. Some of them are smaller. So we look here, this is a given neuron, and neurons are fibrous structures. They have these long extensions, and they have an area from which they receive information. This is your dendrite, and that's also listed here. Dendrites are going to receive input. Information is then sent away into the next neuron via the axon. So this region down here, this is your axon that sends the information. You can tell by looking at this that they're incredibly branched, and they can communicate over very long distances. And then if we zoom in on the axon, there's going to be the end of an axon known as the axon terminal. And if we zoom in on the tip of this axon here and zoom in, you can see this axon terminal here very clearly, and we're going to look at that much more later. And you can see how that axon then can send information to the next dendrite. Remember, we said dendrites receive information, so this here could be the dendrite of a neighboring neuron. And then if we were to look at the space that includes the axon and the dendrite and the space between it, we call that a synapse, that junction. And then if we look at just this region here between the two neurons, we would call that the synaptic cleft. And we're going to go over this very clearly later on. But you can see that the way that these neurons communicate is not by physically touching. There's actually a gap there between them, and that's really important. They never physically touch. Instead, what you see happen is that you have release of what is known as neurotransmitters into the space, and then they released from the presynaptic cell. So the axon would be considered the presynaptic because it's before, sends the information into that space, and then it can bind to receptors on this postsynaptic membrane and then cause a response, and we will get into the nitty-gritty of this in the next bit. So I'm not going to read through this all right now because we're going to go through this all very slowly, but I did write this all up so at least you had it written here for reference later on. So again, your neurons are going to vary in size. Some of this I already told you before. You have a very long motor neuron that goes from your spinal cord all the way to your big toe, which is several feet long. And then you have interneurons that are really short. So they can vary quite a bit. They can also vary in terms of the number of extensions that come from it. Really, we're not going to get into all this, but certainly there are courses where you could take more in-depth and learn more in-depth knowledge. And then the cell body is this region where the nucleus is going to be located, and it's going to be positioned differently in all of these different neurons. But the other thing I wanted to point out here is that your brain is actually comprised of more of a different type of cell than neurons, and what your brain is mostly comprised of is actually glial cells. And these are cells that help to support the neurons. They don't actually transmit any electrical information, but they can help surround the neuron and provide it with both physical and nutritional support. And we'll talk a little bit more about how they insulate neurons later on. You can see this here, this insulation, and we'll talk about that later. So again, your neurons can receive information from many other neurons and send information to many other neurons. There's an extensive branching that takes place. Most neurons can form anywhere from hundreds to thousands of synapses. So there's some very complex circuitry taking place here. And they also can communicate with other cells that might receive electrical stimulation, such as your muscles, which we'll talk about more later. Secretory cells we don't really get into in this course, but certainly muscle cells we'll talk a lot about. Although I take that back. Secretory cells we will talk about more with the endocrine system later on, so that is actually also both of those we'll talk about. So before we move on to the next section, I think it would be useful if I do a quick review of membrane transport. So bear with me if this is something you're familiar with, but I want to make sure everyone's on the same page. I've done this before, but now I'm going to do this in a little bit more depth. So we know that your plasma membrane is semi-permeable. So let me just briefly sketch plasma membrane. So your plasma membrane is made up of many components, but the basic one is your phospholipids, which is what I'm drawing here. So each individual one of these guys I'm drawing is a phospholipid. And I'm just going to draw a membrane. You're going to note that it's a bilayer, bi meaning two, because you have a double layer of these lipids that are facing toward each other. All right, so I've drawn quite a few of them here. So let's just review some basic premises of how what their properties are. So these tails in the middle portion, these are the portions that are hydrophobic. They're afraid of water. So hydro meaning water, phobic meaning fear. So these are the portions afraid of water. So anything that is not water, actually I take back, anything that has properties similar to water have a hard time getting through here. And I'll review a little more in a minute. And then we have these head portions, right? These head portions over here, and these are hydrophilic. Right? So both of these guys here, they're hydrophilic. And so it should make sense then that the inner portions that are hydrophobic face inside, because everything outside here is water-based, right? So all this stuff out here is water-based. Inside of your cells are water-based, outside of your cells are water-based, hence they interact with the hydrophilic portions. And then the parts that are hydrophobic, the parts that don't like the water, are on the inside, staying away from the water. They just pack in tightly together. All right, so we've got all that. That's great. Now let's think about the molecules that are hydrophilic versus hydrophobic. So things that are hydrophilic have properties similar to water. So water, if you remember from the plant portion, water is comprised of a polar molecule. So we have H2O, and you basically have oxygen, right? Water, hydrogen, hydrogen. And we know that oxygen loves electrons, so it pulls those electrons in more tightly. And they're more clustered toward the oxygen. And that means these electrons that are negatively charged are kind of hanging out closer to the oxygen. So we give that a partial negative charge. And then the hydrogens have lost some of that, so we give those a partial positive charge. And that creates this polar molecule with a partial positive, partial negative. Anytime we see anything with charges, we always want to associate them with water. And that's basically what we've drawn here, these partial charges. So things that are charged are hydrophilic, right? So a lot of ions we're about to talk about in neurobiology. So we have sodium ions, they have a positive charge. Potassium ions have a positive charge. Chloride has a negative charge. Calcium has a positive charge, right? All these ions are things that are not going to easily get through the lipid bilayer because they're hydrophilic, properties similar to water. So then how do they get through the lipid bilayer? So we're going to have different types of channels. We can have channels that are in the membrane. So let's throw a channel here in the membrane. And then let's add some more lipids around it. And there are different types of channels, and all of them are very specific. So let's say that this channel here, this one is specific for sodium. Let's call it a sodium channel. And so this channel would only move molecule sodium. Anything else it can't move. We might have another channel hanging out here. Let's say there's one over here. And this one over here might be specific for potassium. So then you could have a potassium channel. So you only can get things that are charged across the lipid bilayer if there is a channel that allows for its movement. Okay, so that's just a general rule. All charged ions, right? So ions by nature are charged. So all charged molecules, but ions in particular, these are single elements that have a charge to them. All charged ions must move via a membrane protein. Great, so now let's talk about the different types of membrane proteins. I'm going to start a new page. Okay, so now we know we have a lipid bilayer, and I'm going to just draw it as a line right now, and that lipid bilayer has different types of membrane proteins. So there are some membrane proteins, let's draw this one here, that move things that are charged. All right, so let's say it's a sodium ion, and let's say we have tons of sodium on this side, right? Lots and lots of sodium over here, and very few sodiums over here. We know that molecules like to move along their concentration gradients. So if it's going to move through this channel from high to low concentration, so it moves through channel high to low concentration. We call this facilitated diffusion. And this would be passive transport. And the reason it's passive is because it does not require energy, so no ATP required. So moving from high to low concentration, no ATP required. That is passive transport. And when we talk about a type of passive transport that requires a membrane protein, we call it facilitated diffusion. Another type of passive transport could be simple diffusion. As an example, use a different color here. Oxygen is sort of an exception to a rule. It has some polar properties to it, but it actually can easily move across a lipid bilayer without the help of a channel. So it will just, if there's a couple over here, it'll move in this direction, but it does not require a channel. This is still passive transport, but we call this simple diffusion because there's no membrane protein required. And there's still no ATP. Right, so neither of these require ATP. Lastly, and this is the one we're going to spend more time talking about, there could be a situation where you have, let's say, tons of sodium over here, right? So we know there's a ton of sodium over here and not a lot over here. And let's say you want to keep moving sodium in this direction. How do you do that? So then you need a channel, a membrane protein. It's not just a channel though. You need a membrane protein that requires ATP. So we're going to use energy, and then we're going to pump sodium in this direction. So now we're going to pump, let's say, sodium ions in this given direction, and we call that active transport. So if I were to draw a nice line down the middle here, right, on the left, these are both passive, and on the right, we have active. So now that we have all that review under our belt, let's discuss membrane potential. So neurons have to communicate. We've talked about that. So the question is, how do they communicate? And they communicate via electrical charge. That's the only way that they're able to communicate is going to be changes, temporary changes in electrical charge. And so then the question is, well, how do they know there's a difference in electrical charge? And that's because they're always charged to begin with. So neurons have a property known as membrane potential. And this is a difference between the number of charged ions inside the cell versus outside the cell. And if you look at this image here, you can see this lipid bilayer, right? So this is the hydrophobic lipid bilayer, and you can see that ions are non-equally distributed on both sides of this membrane. And the reason that they're non-equally distributed is due to a pump that's working to put them in certain places. But what I first want to point out when you look at this is that, yes, there's a difference in charge. That difference in charge can be measured in voltage. We call this the millivolts usually is how we measure this. And the resting membrane potential, so a neuron that's not sending any sort of activity, is going to range anywhere from -40 to -85 millivolts. So the inside of the cell is always more negative relative to the outside. And that's written right here for you. And that number, oftentimes we just write as -70, sort of to give an average. The resting membrane potential, you can see here, is at about -70. And this can be measured with voltage clamps. So if you look here, they're showing you this reader is measuring it with these little clamps that are pinched on the membrane. And scientists can actually do this. You can stick electrode into a neuron and measure its voltage. So then the question is, how is this voltage created? And it's created via a pump. So there is a pump working very busily inside all of your neurons, and this actually uses most of the ATP in your body, and it's called the sodium-potassium pump. So every time it pumps sodium and potassium, it uses one molecule of ATP, and it is always going to pump 3 sodium ions out for every 2 potassium ions it moves in. Therefore, it's moving more positive charge out than it's moving in, so you end up with a net negative on the inside of the cell. And here you can see this sodium-potassium pump. I will draw this for you over and over again. It is always going to pump 3 sodium ions out. This is out because this is extracellular over here. And for every 3 sodium it pumps out, it's then going to pump 2 potassium in. And that's going to create a net negative charge on the inside. In addition, there are potassium leaky channels, which you can see here. And these channels, these would be considered, remember, passive transport, no ATP is required, but it's facilitated diffusion because there's a channel helping the movement of these potassium ions. And these are leaky to potassium. So they're specific to potassium. And since this pump is constantly moving potassium inward, you end up with excess potassium on the inside, and some of this will slowly leak out in this direction, moving its ion concentration, and therefore positive charge is even more leaving, and that gives you an even more negative charge inside. What I don't talk about in this class is how this actually is a gradient that's more than just a chemical gradient. This is an electrochemical gradient. So it's both charge-based and chemical-based. The chemicals would be the sodium and the potassium. The electro would be the charge that comes with them. So this electrochemical gradient. So you have to think about equilibrium in terms of both ions as well as charge. So even though you're pumping in tons of potassium, all of the potassium can't rush out through that leaky channel because it won't work in terms of the charge distribution. It'll be repelled. So a little bit can leak out, but not all of it. And that's again because of the electrical component of this gradient, not just the chemical component. We'll look at this in class together. Okay, so again, we talk about this resting membrane potential, and actually all cells are going to have some resting membrane potential. Sodium potassium pumps work very hard in all of your cells, but only neurons can respond to a change in membrane potential. And their ability to respond to change makes them excitable. They can transmit this change, and their ability to transmit is due to a specific type of channel found only in neurons as well as other excitable cell types such as muscles, which we'll talk about later on. And their ability to respond is due to what are known as voltage-gated channels. Voltage-gated channels are ion channels that can open or close in response to a change in voltage. So you can see here, for example, this particular channel is currently closed. So you can see the closed position over here, and then if you look over here, this channel is now open. So what caused that opening? Well, it turns out that this protein changes shape in response to a change in voltage. So here in picture 1, it has one given shape, and then you change the charge around the protein, and that causes the protein to change shape, and then you end up with number 2, and now it's open. Protein changes shape in response to change in voltage. And there's a specific voltage at which this protein changes shape, and when it does that, it opens up. We would call this passive transport. There's no energy required here, just a change in voltage, so no ATP. And we would call this facilitated diffusion because there's a membrane protein involved. And what we'll talk about more and more is that these voltage-gated channels only open at a given threshold, and then it's all or nothing. They either open or they don't open. There's no sort of open or sort of closed. And the number, there's a specific voltage at which they change, and we call that threshold. So there's a threshold voltage at which they change shape, and then they open. And also, like all other channels, they're specific. So we might have sodium voltage-gated channels that can move sodium, as you see here. There are also potassium voltage gated channels that only move potassium, right, et cetera, et cetera, et cetera. We'll look at this together in class. So now we're going to talk about how neurons transmit this change in electrical potential. And they do this through a process known as an action potential. And an action potential is going to be a rapid and temporary change in membrane potential. So what we're looking at here in this picture on the right would be an example of an action potential. There are 3 phases to an action potential. The first phase is, and I should also point out that this is something that happens in a very localized region of the neuron. We'll talk about how it's transmitted down the length of a neuron. But if you're looking at, say, I'm a terrible artist, but here are my dendrites, the cell body, right, this long axon, and then the axon terminals that branch at the end, and we're measuring where an action potential is taking place. We're measuring just this one little local area right here. So there could be an action potential happening right here. There could be an action potential happening here. There could be one happening here. It's happening at very discrete localized regions of the neuron, and it can be transmitted down the neuron, so it can cause the next region of the neuron to also have this temporary change in membrane potential, but it's not like the whole neuron is actively undergoing this at the exact same instant. It's a very local change that's taking place, so you could measure a given region of the neuron. So if we were to measure a given region of the neuron and see it's undergoing this change, we would note 3 phases. The first phase would be the depolarization phase, and that's what you see in this part right here, right? So it's increasing in charge. So it's becoming less negative. It's moving from this negative 70 all the way to 0, which is neutral, and then even beyond that and to the positive number. So it's becoming less negative and eventually more positive. Then eventually you go through the second state, and that is repolarization. This is where you return to normal. So now you can see that this is decreasing in charge, returning to the resting membrane potential. But then you reach phase 3, which is hyperpolarization, and this is the phase in which it then overshoots and becomes too negative. Eventually, you then repolarize, moving in this direction again, and then eventually reach the normal resting membrane potential. And we're going to talk about this in a lot of detail and how it's created. So I'm going to say that for a cell to have an action potential, it must become sufficiently depolarized to reach threshold potential. And remember, threshold potential, this threshold relates to when the voltage-gated channels open. So voltage-gated channels are going to open at a given change in membrane potential, and that is going to be referred to as threshold. So remember that your resting membrane potential is around -70, -80, give or take. And again, this is a give or take number. Your threshold will be, let's say, -55. So your normal resting membrane potential, you're hanging out, -70, -80. Something causes the membrane potential to change. We'll talk about what that something is later on. But let's say it changes and now all of a sudden we're at -55. Boom, voltage-gated channels are going to open. So action potentials that occur due to this happening are all or nothing. You're not going to sort of have a cell reaching threshold. Maybe I'll have an action potential. No. If you reach threshold, the action potential will happen. Boom, done, no question. And it happens because step 1, remember that you have step 1 is depolarization, and that depolarization is due to a sodium voltage-gated channel opening. So you have reaching threshold. The first thing that happens is your sodium voltage-gated channels open, and they're going to allow sodium to rush in along their gradient, causing further depolarization. So again, if you remember, you have a sodium-potassium pump. It's pumping 3 sodium out for every 2 potassium that comes in. If all of a sudden you have a voltage-gated channel specific to sodium that opens, then all of this sodium can rush back in, and then that positive charge rushes in, and then you're going to have depolarization as the inside of the cell becomes more positive. So again, that was the depolarization phase. So here you can see depolarization, right? That is due to sodium voltage-gated channels that open. Voltage channels open, But then when you reach this peak point, that is where sodium closes, right? So sodium voltage channels close. At the same time those close, you have other voltage gated channels that open up here, and those are your potassium voltage channels. They're also voltage gated channels, and they open in response to that change in voltage. They're just a little slower to open than your sodium channels were. So they responded to that same initial change in voltage. They're just a little slower to get started. Once they open, again, remember you have a sodium potassium pump, right? 3 sodium out for every 2 potassium in. And if you open a voltage gated channel for potassium, where is potassium going to go? It's going to leave through that voltage gated channel. And as that leaves, it's going to cause positive charge to exit, and you're going to restore the inside of the neuron to a more negative state. However, they tend to stay open a little too long, and that's what this hyperpolarization is due to. So again, you can see this last part, the hyperpolarization. That's because those potassium channels stay open a little too long. So we just kind of move past the resting potential and you become hyperpolarized. But eventually, right, at this point right here, that is when your potassium voltage gated channels close. So we stop losing potassium. And now everything's closed. The sodium voltage gated channels, they closed way up here. And now you've got your, sorry, I hit a button. And now you've got your sodium channels closing up here. So everything's closed. And then you are going to end up having nothing working anymore except for your everyday normal things that allow the resting membrane potential to reestablish, right? Your sodium potassium pump, the leaky potassium channels, everything's working normally now, and then you just restore resting membrane potential. So just to review one more time, right? You're hanging out at resting membrane potential, and then something happens that brings you to threshold, right? So now we're hanging out at threshold. We'll talk about what that something is later on. And then boom, you reach threshold. This is all or nothing. Things start opening. First thing to happen is right here when you reach threshold, that's when your sodium voltage gated channels open. That causes sodium to rush in, and that's going to cause you to depolarize, right? This is your depolarization. Then you reach a certain point at which the sodium voltage gated channels close. So up here, the sodium closes. And at the same time, those potassium voltage gated channels, they were a little slower to open. Now your potassium opens. That causes potassium to rush back out of, potassium is going to leave, so potassium is now exiting, and as that does that, it is causing positive charge to leave, and as positive charge leaves, the cell gets more negative, but they stay open a little too long, and they hyperpolarize until you get to about here, and then finally the sodium closes, and then voltage gated channels are no longer open, and the only thing left that's working are your sodium potassium pumps and your potassium leaky channels, and then you restore normal resting membrane potential. We'll look at this in class together. We'll also look at this in class together. So I think I've made this clear, but I just want to make it super clear. Action potentials are all or nothing, right? There's no sort of having an action potential smaller, larger, no, they're the same. So for any given neuron, your action potentials are identical in both magnitude and duration, and they happen in very local regions of your neuron, but then they can cause a neighboring area of your neuron to then respond and have that same action potential. So they get propagated down the length of the axon, and we'll talk about how that happens. So I'm going to have lots of movies and pictures to show this, but I have words here to show how this is propagated. So let's just say you have a long axon, and remember there's a given region that's having an action potential. So let's just say this region right here is having an action potential, right? So sodium voltage gated channels open, and we know sodium rushes in. So in comes all that sodium due to depolarization. Remember that ions can diffuse, so they're going to start kind of moving in both directions. And sure enough, as it starts moving in this direction, it will then trigger a change in membrane potential in a neighboring area that can cause it to have an action potential. So now, all of a sudden, while area number 1, we'll call this number 1, is busy having its action potential, the influx of sodium is causing the neighboring area to then depolarize enough to reach threshold, so that then area number 2 has its sodium voltage gated channels triggered due to a change in voltage, and then sodium's going to rush in here, starting an action potential here. So more sodium rushes in, and then it starts diffusing, and then sure enough, area number 3 is now triggered, etc., etc., etc. So this is a self-propagating process. It's an example of positive feedback because the action itself causes it to happen even further, right? Area number 1 causes 2, causes 3, the next one opens until you have this propagating down the length of the neuron. It actually only moves in one direction. We'll talk about why that is later on, but it does in fact only move in this direction. When I indicated that it diffused in both directions, that's true, but this area before just finished its own action potential and it cannot have another one right afterwards, so this will not be triggered, and we'll talk about why that happens later on. Oh, the dog's barking again. Okay, I think she's done barking now. So again, action potentials move in only one direction, so they're going to be propagated in this direction. That's why the arrows are moving this way. And you can see here how in this particular image you have sodium voltage gated channels that are open right here in this area, right? So remember, the inside of the neuron is usually more negative than the outside. So you can see over here, it's nice and negative on the inside. So this is at rest. Over here, it's negative, so you can tell that's at rest. But then this middle area, you can see how it's positive on the inside, and that means there's an action potential happening. And then if you look at what happens in step 2, that area has moved further down the neurons. So now this area is undergoing an action potential, while this area over here is at rest and this area over here is at rest. So you can see how it just shifted toward the right. One area triggers the next to start having that action potential. And again, that's because when you have those sodium ions that rush in, right? So here you can see those sodium ions that are rushing in, they diffuse down into the neighboring area, causing it to then have an action potential, and you can see that here. So that you can see that while this area, we'll call this area number 1, and this 2. So area number 1 is now busy repolarizing, and you can see that because now you've got the potassium rushing out to reestablish charge. While it's busy repolarizing, the next area, number 2, is now busy depolarizing. So again, this is a one-way propagation of the signal. They're moved down the length of the neuron. But again, the response is all or nothing. You just have given areas that are experiencing that potential change, and then the next area does the same thing. They can't go in the other direction, and the reason is, I said the previous area had just finished having its own action potential and can't start up a new one so quickly. And the reason is that sodium voltage gated channels need periods of time to reset. And while they're busy resetting, we call them in the state of this refractory period. Oops, did not mean to move that around. So here you can tell they're in this refractory period, so they need a short period of time to reset. But the ones that haven't yet experienced the action potential on the other direction, they're ready to go, and so they can then experience that process while the other area is busy resetting. Therefore, it has to move in one direction. And this transmission of an action potential actually moves very rapidly. And part of the reason that allows for this rapid movement is that your neurons are insulated, just like you would have any electrical wire insulated to allow more rapid movement so you don't lose ions to the outside. And the way your neurons are insulated is through special types of glial cells. We talked about them earlier. And these glial cells are going to provide what's known as a myelin sheath. They're going to surround the axon and insulate it. And this is found in vertebrate neurons. And this will help to increase the speed of this transmission. And then you have areas where you have an absence of that sheath, and you can see that the blue is the insulation, and then this area here is that absence. We call the area where there is no insulation a node of Ranvier. And that node is an area that's exposed without the sheath, and they're going to alternate between areas with and without insulation. And the areas that don't have the insulation, that's where your voltage-gated channels are concentrated, and that's where you're going to have a boom, that rush of action potential, and then the diffusion that occurs rapidly will occur down the length of the neuron where the insulation is, and that won't be able to escape because of the insulation. Therefore, it's a very rapid transmission, then allowing voltage-gated channels in the next node to be triggered. So the action potential sort of jumps from node to node, and you can see that in this picture here where they have the, well, actually I showed it going in this direction, but let's say it's going in this direction because actually that's how it would go because it comes in at the dendrites. So it jumps here, and then it'll jump here, and then here, and then here. This is a great video. It's from a different textbook. You can see here it's from Pearson, and we will look at that in class together, but it is found on Blackboard if you want to take a look. So you'll notice that in that previous picture I was drawing, and then I said, oops, I drew this in the wrong direction, because information's always received at dendrites and then is sent down the length of the axon toward the axon terminal. So we're going to talk about how you have neurons communicating with each other. So let's say that we call this a presynaptic neuron. So here's a neuron that receives information at its dendrites. These are the dendrites. That information is processed at the axon hillock, which is really at the base of the cell where the cell body meets the axon. And all of that information gets processed, and then right around here is where you first start to have a concentration of voltage-gated channels, and that's where the action potential becomes triggered. And then it will be sent down the length of the axon. Remember, it only moves in one direction. And then it'll reach eventually the base of the axon. And when you get there, that's where the synapse is going to be located. Neurotransmitters will be released, communicate with dendrites on the postsynaptic neuron, and then it will have its own action potential that starts again here and is sent down its axon. And we're going to momentarily focus a little bit more on this area and how that transmission takes place. So again, I'm going to review this with a picture momentarily, but I always write the steps out for you so that you have this for reference later on. So now I'm going to go through in this picture exactly what I wrote out for you in words before. So remember, you have your neuron. So let's draw my neuron here, right? It's got the dendrites and the cell body, and then the axon, and then the axon terminal. And what we're looking at right now is we are zooming in on the axon terminal, right? So the information was received in the presynaptic neuron at the dendrites. It's summed up here in the axon hillock. It's sent down the length of the axon. It reaches the axon terminal, and now we're zooming in on this region right here and how it is communicating with a postsynaptic neuron. So let's say there's some dendrites over here, another cell body, right? So we're zooming in on this region and how you're communicating between the presynaptic neuron and the postsynaptic neuron. So we're in neuron number 1. This is the black one I drew over here. The axon terminal, the axon has had the action potential move down its length. We're now reaching the axon terminal. And now in the axon terminal, there's a new type of voltage-gated channel found only here that's not found in other areas of the neuron. And these voltage-gated channels are calcium voltage-gated channels. Turns out that action potential, when it moves into the axon terminal, will cause calcium voltage-gated channels to open. And there's more calcium outside of a cell than inside, so that causes calcium to rush into the cell. So now we have tons of calcium flooding in the cell. Well, there's a bunch of steps that cause this to happen, and I'm not going to go into them all. There's some snare proteins and other factors involved in this process, but basically, when calcium floods the cell, it is going to cause these vesicles that are stored at the base of your axon terminal. It's going to cause them to fuse with the membrane. So you can see here one of these vesicles in the process of fusing, and it's going to dump all of their components into this synaptic cleft. And so these vesicles stored at the base of your axon terminal, they are called synaptic vesicles. And these synaptic vesicles, which I'm sure is written somewhere here, stored in vesicles, right? So these are your synaptic vesicles. They store neurotransmitter. And when they fuse with the membrane, as shown here and here, all those neurotransmitters that are stored up in the vesicles, they then get dumped into this space. So you can see all of these guys, these little blue squares, they're now exiting and they're in this synaptic cleft. Then those neurotransmitters can bind to receptors on the postsynaptic membrane and cause the postsynaptic membrane to itself then possibly reach threshold itself. So in this example, I'll describe to you how this one works, and we'll talk about it more when we get to muscle, because this example is specific to muscle. So acetylcholine is the example shown here as a neurotransmitter. Acetylcholine is a type of neurotransmitter that binds to a ligand channel on the postsynaptic membrane. So you can see here that these are channels, and you can see this one's closed and then this one's open. And when it's open, it allows sodium ions to move through. So this particular type of channel is specific to sodium ions. But you'll notice that when it opened because acetylcholine bound to it. So this is not only a channel, but it's also a receptor. And we call these ligand-gated channels. So you might remember from last semester, we talked a lot about receptors that bind different molecules, and the general term we use for any molecule that binds a receptor is a ligand. Sorry about all the vocab, I didn't make it up. So you can see there that just in general, a molecule that binds a receptor we call a ligand. So these are ligand-gated channels, so they're receptors that must bind to a ligand, and when they do, it causes the channel to open to whatever ion it is specific for. And it turns out in this case, they're specific for sodium. So when these ligand-gated channels open, sodium rushes in. And what do you think happens when sodium rushes in, right? Sodium is going to lead to depolarization, just like it does in the other cases I can't spell. That's supposed to say depolarization. Depolarize. And then you're going to cause this postsynaptic membrane to reach threshold. And we'll talk more about that. So everything I said previously is now written here in words for you. So many neurotransmitters, not all, but many, are going to function as ligands that bind receptors. And these receptors are called ligand-gated ion channels. And in response to the ligand binding, the channel will open. Ions will enter along its gradient. So all the different types of ions, some ions when the channel opens will move into the neuron, some will move out, depends on the neurotransmitter. Sodium happens to move in. Neurotransmitter's chemical signal is then transduced to an electrical signal, right? So you have a neurotransmitter that binds to a channel, let's say in this case, and then causes an ion to move, and that ion movement leads to a change in membrane potential in the postsynaptic cell. Every neuron makes only one type of neurotransmitter. So all your neurons are types of neurons that make given types of neurotransmitters. And while one neuron can only make one type of neurotransmitter, neurons can respond to many different types of neurotransmitters. That's written here. And we'll talk about how a neuron is able to receive lots of different information from different neurotransmitters and then figure out what it's supposed to do. We'll talk about this one in class. So remember I said that a postsynaptic neuron can receive lots of information from different neurotransmitters. We talked about acetylcholine. Acetylcholine causes a sodium ligand-gated channel to open that causes sodium to flow in, and that causes depolarization, which makes it more likely to have an action potential. And because of that, we call this an excitatory postsynaptic potential because it's likely to make the next cell depolarize and therefore have an action potential. But there are other types of neurotransmitters that cause other types of channels to open. And in this case, it could cause a more hyperpolarization, right? If you have more hyperpolarization, then it's less likely an action potential will happen. And as an example, if you have potassium rushing out, that will cause hyperpolarization, or chloride is negatively charged. If it rushes in, that could cause hyperpolarization, and that would cause what's known as an inhibitory postsynaptic potential, and it's inhibitory because it makes it even more hyperpolarized, and that will make it even more difficult to reach threshold because you're moving even further away from that threshold voltage. But neurons can receive both excitatory and inhibitory information at the exact same time, and you can imagine when that happens, they cancel each other out, and then nothing happens. So neurons have to sum up a lot of information and then figure out what they're going to do. So again, neurons receive a lot of information. They make hundreds and thousands of synapses. And in fact, if you look at this neuron here, so here you can see the cell body and all of these extensions. These are your dendrites that are receiving information, and every single dot here, every yellow-green dot along the length of these dendrites is a synapse. That's crazy, right? Hundreds of thousands of synapses, and all of this information has to be summed up. And the summation occurs, as I mentioned, in the axon hillock. That's sort of at this base of the cell body, right where the axon projects from the cell body. And you can imagine that if inhibitory and excitatory signals cancel each other out, then nothing's going to happen. But if you have many excitatory signals right after the other, they can sum up and then make it likely that that neuron will fire an action potential. So remember, the whole thing here is, are we causing this neuron to send a signal or not? And remember that sending a signal is all about having an action potential or not having an action potential. And how we have an action potential is by reaching threshold, right? So we're hanging out here at resting, and then the question is, are we going to increase charge, depolarize enough to reach threshold? And we do that by having an excitatory potential. So remember, you can see lots of different information. So let's say in this case, we're having a temporal summation. Temporal summation would be multiple excitatory signals being received simultaneously from a given site. So here you have a single synapse that's boom, 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 sending off sending off excitatory potentials. And one after the other, that one neuron sends a signal, and then you might reach threshold due to that signaling. However, other types of summations could be spatial summation. Maybe you have this excitatory and this excitatory and this excitatory, all of these different excitatory neurons sending information, and all of that one after the other can then cause you to reach threshold and then have an excitatory potential. Or you could have an excitatory signal and an inhibitory signal, and then they cancel each other out and you get nothing. Right? So lots of different ways that this information can be summed up. This is another great video discussing everything I just talked about, which we can watch together in class, and it is also available on Blackboard for you. We will look at this in class together. So this last section, I'm going to be honest, we do not spend a lot of time on this. A lot of this is just memorizing some facts. I try to give you the basic concepts here, but I will tell you that if you are planning on taking the MCAT at some point, there are bits of this that you should memorize, and I'll try to point out what those things are in case that's something that's relevant to you. So briefly, I'm just going to talk about overall the big picture. How is your nervous system comprised? You have a central nervous system. This would be your brain and your spinal cord. This is sort of where the processing takes place. And then you have your peripheral nervous system that extends from your central nervous system, and your peripheral nervous system is where all the information is going to be received and then sent out. So in summary, how this is going to take place, you're going to have sensory information from receptors in the peripheral nervous system that is sent to your central nervous system, that is then processed by your central nervous system, and then that can be sent back to appropriate parts of your body to cause actions to take place. So again, your central nervous system does its thing, it processes, but then when we talk about the peripheral, that's anything outside of your central nervous system, you're going to have both voluntary and involuntary components. Things that are voluntary are considered somatic. This is sort of your conscious response to things, your ability to respond to the external environment. And then we're going to talk a little bit more about things that are unconscious, involuntary, and these are somatic. And these are things that obviously regulate your internal body functions, and they can be divided even further into your parasympathetic and your sympathetic branches. They help to monitor internal functions. And this is where I'm telling you that I am not going to ask you to know all this, but I will tell you if you're studying for an MCAT, they often like you to know the difference between your parasympathetic and your sympathetic system. And your sympathetic tends to stimulate things. Your parasympathetic in general tends to slow things down. I'm not going to get into this in this class, but again, if you're taking an MCAT, there's some MCAT books that'll tell you what you need to know and what you don't need to know. Okay, so on our last slide, we're going to talk about a specific type of response that actually does not involve your central nervous system. Or specifically, it involves your spinal cord, which is part of your central nervous system, but it does not involve your brain, so it's not processed by cognitive functions at that level. And these are your reflexes. So reflexes are involving very rapid responses, and oftentimes they're involving things that would help to assist your body in a rapid response that would be needed to help you sometimes even avoid danger or threat. So in this example shown here, this isn't a danger or threat. This is just a very simple reflex that can be tested in your doctor's office. They use a strike force on your knee, and they hit a particular location where there are stretch receptors that open due to a response and that pressure from that mallet when it hits your knee. And when that receptor opens, it causes an influx of ions that then will lead to a action potential. So then this neuron that receives the information from the stretch receptor, the sensory neuron will send information back to your spinal cord where it will be processed, and not really even processed. It just is sent, right? The information is sent along. So it's sent then via an interneuron, which is this short neuron that is in between. And then that interneuron will synapse with two different types of motor neurons. One of them will communicate with your extensor muscle, and one will communicate with your flexor muscle. And because we'll talk about this in the next section or a couple sections from now, when your muscles work, they work opposite of each other. So one will contract while the other one relaxes. So when you kick, one of these motor neurons is telling your extensor muscle to kick while it's telling your reflexor muscle to relax. And this very short circuit, again, does not involve your brain in terms of processing the information. You simply have a sensory neuron. That sensory neuron will synapse with your spinal cord, and then that interneuron in your spinal cord will then synapse with two of your motor neurons that will cause your leg to kick. A more interesting, I guess, example of this is one I often give, where I was working in a lab years ago, and I was cutting a gel. And yeah, silly me, I was cutting this gel with a razor blade, brand new. I'm pushing it down with my finger, and I'm trying to figure out why this gel won't cut. And then all of a sudden my finger pulls back, and about 2 seconds later my brain processed that information, and I realized I was holding the razor blade upside down. Oops, that's what happens when you don't have enough sleep in grad school. So what was interesting about that response, and why I tell the story though, is that my hand pulled back about 2 seconds before, or even like a split second really, but it was a split second before I processed the fact that it hurt. And that's because of that reflex response. So something happened in my finger that sensed this information, which was sent to the spinal cord and rapidly back to my finger, which caused my finger to pull back because of this danger that it had countered. And then later on, that information was sent to my brain, which processed it, and then I was like, ouch, right? So 2 different responses going on there. One is the reflex that causes you to draw back. The second one is, yes, information is sent to your brain and you process it, but it happens at a more delayed fashion. So this is why the reflex itself is a very, very fast response, because it doesn't actually cause the information to send to your brain. Brain function requires a few extra seconds or a split second longer. And so that is what the difference is there. So a lot of information in this one, which is why we're going over it in the course of 2 days. So basic information about your neurons, how your action potentials function, and then what happens at the synapse between your pre and your postsynaptic neurons.